Welcome everyone to the Bat Talk, hosted by North Sydney Council, where we have our guests, Dr. Nicholas Wu and Tomás Villada-Cadavid. I hope I did that name justice. Sorry if I didn't. Who will be talking about white nose syndrome. I would just like to just a little bit of background. Western Sydney University approached us to do a study on white nose syndrome as part of one of our sites, the Coalota Tunnels. So some locals might know that we have a population of large bent-wing bats at this location for part of the year, and these academics were interested to look at this habitat and to look at the habitat there, whether it was how it related to white nose syndrome, which our guests will speak about in a lot more detail in a minute. I'd just like to ask everyone to use the chat function to ask questions. We won't. No one will have audio during this talk, so please ask questions as we go along. And and those questions will be answered at the end. I'd just like to have a little bit more formal introduction for our guests. Dr. Nicholas Wu joined the Hawkesbury Institute for the Environment in February 2022 as a research fellow within the Plants and Animals and Interactions theme. Nicholas earned his Bachelor of Science from the University of Waikato, New Zealand, followed by a Bachelor of Science in Zoology and a PhD in Ecology Physiology at the University of Queensland. From 2019 to 2021, he was appointed as a postdoctoral research associate at the University of Sydney and is currently a postdoctoral research fellow at Western Sydney University. At the Hawkesbury Institute of Environment, Nicholas is working on an ARC linkage project under the supervision of Dr. Christopher Turnbull and Associate Professor Justin Welbergen. The project aims to develop the predictive models of vulnerability to white nose syndrome, WNS, for populations of Australian bats by gaining critical information about their risk of exposure and sensitivity to mortality from developing winds. Beyond research, Nicholas is also experienced with digital illustrations and graphic design, where he incorporates them to disseminate his research to both the scientific and public community. And our second speaker, Tomás, who's working on this project with Nicholas. Tomás Villada-Cadavid is a PhD student at the Hawkesbury Institute for the Environment. He's working on this project, Vulnerability of Australian Bats to White Nose Syndrome. Tomás has worked with bats since 2015. He first earned his Bachelor of Science in Biology at the University of Antigua in Medellin, Colombia, from where he assessed bat diversity in a karstic corridor, and led to the declaration of an area of importance for conservation of bats, recognised by the Latin American and Caribbean Network for the Conservation of Bats. Later on, Tomás moved to Germany, where he studied in biodiversity of bats. Later on, Tomás moved to Germany, where he studied in biodiversity and a collection of management at the Technical University of Dresden, and had the opportunity to collaborate for his master's on a thesis with the Natural Science Museum of Groningen's bio-research group, researching bat diversity and how it correlates to the environment and structural traits of caves and surroundings during the swarming season in Catalonia, Spain. Tomás is currently enrolled as a PhD student at the University of Western Sydney, where he aims to generate spatial explicit models to assess bat vulnerability to white nose syndrome. Well, welcome, Nicholas and Thomas, please take it away.
Thank you, Andrew, for the introduction. I'm going to share my screen with you and we'll get started. Right, can everyone see my screen? Can you confirm, Andrew?
Yes.
All right, thank you everyone for coming today. So what we're going to do is we're going to talk a little bit about the project that we are working on at the moment for my PhD and Nikki's postdoc. This is about vulnerability of wait a second, I want to hide this video panel. All right. So we want to talk about the vulnerability of Australian bats to white nose syndrome. And this project, it's a part of an art linkage grant from the government and part of the Western Sydney University and the Bats Lab. Oh, my screen, now it's working. So I want to talk a little bit about bats first before I start talking about the project itself that Nikki is going to introduce in a little bit. So first of all, bats are mammals compared to what people might think. Some people think that bats could be birds or other animals, but they are actually mammals. Like humans, they are warm-blooded, they're hairy, they don't have feathers like birds do, and the youngs feed on milk. That's a common trait of all mammals. You can see here how bats are organized on the phylogenetic tree within the mammalian groups. And you can see how bats are closely related with carnivores like dogs, cats, and then they're also related to horses, llamas, and dolphins and cows. What I wanted to point out here, it's how bats how they can also do all things that mammals can do. They can see, they can hear, they can smell, and they can feel as well. They have an outstanding way of moving around, which is echolocation. They emit some ultrasound, and that ultrasound comes back when it bounces against objects, and they can kind of sense like a radar what they have around, because they do all of this. They fly at night, and it's when there is not good light conditions, so they use the ultrasound to see. There is a misconception that bats can't see, but they actually can. They do see. For instance, flying foxes, actually, they don't have echolocation, and what they do is they rely on their eyesight to fly in the evenings, and their smell to go to the places where they feed, finding the trees and food resources. Now I wanted to let you to talk about the word Chiroptera, which is the order in which bats are classified. It comes from the ancient Greek. Chair means hand and pteron means wing. So Chiroptera would mean something like winged hands. And that's what they use to fly. They actually use their hands to fly. You can see here in this image a comparison between humans, birds, and bats. And they all have the same bone structures. They are comparable. You can see how birds compared to humans have less fingers, and all the bones on the wrist have fused together and shortened a little bit, whereas bats have all the fingers that we humans have, and they have elongated, and they support the wing structure that allows them to fly. So it's like a convergence between birds and bats, that they have evolved wings to fly, but it has developed differently in terms of the anatomy, and it works a bit different. On this slide, I wanted to show you how diverse and different bats can be within the group Chiroptera. They can be as small as the Kitti's hognose bat or the bumblebee bat, which can weigh down to 3 grams. And you can see how tiny it is. It's like the size of a thumb. And then we can have huge bats like flying foxes that can reach up to 1.4 kilograms. This is not a good example of how to hold a bat. So please, if you find a bat, never handle bat. Always call professionals to do this. You should never handle bats without gloves and without protection and without being vaccinated, because bats can transmit the virus of the rabies, which is 99% lethal for humans. I want to show you how diverse are bats as well. Here we have over 1,400 bat species worldwide. Australia has a small portion of that diversity, only 81 bats. And New South Wales has 34 bat species. I want to point out here how the tropics are the areas where the diversity is located. Most of the diversity is around the tropics. And you can see how Colombia, where I come from, is one of the most diverse areas for bat species. We have more species of bats there. In terms of shapes and forms and colors, we have here some pictures that can, so you can see how different they can be. This one has like a mohawk, and this is a character that has been selected sexually, which means that it's used to attract females. We have here the hammerhead bat, who has this enlarged snout. It's a fruit-eating bat. This is an insect-eating bat. We have here Macroderma gigas, or the ghost bat. It's an Australian species that you can find in the Northern Territory, and this is a carnivorous species. This one feeds on other bats or other vertebrates. And you can see here Caribula pictata. It's a very colorful bat, orange skin, and very golden fur. Very beautiful animals. These ones can be found in Africa. You have here Lacyrus styrneus. It's a North American bat, also with beautiful colors. You can see the fur has these like white stripes. And we have white bats with yellow skin in the tropics. In Costa Rica, you can find Ectophila alba. It's a bat that roosts in tents. I'll show you a little bit more about this later. They have different food habits. They can feed on fruits, they can feed on nectar, they can feed on insects that they can catch while flying, which is very impressive because it requires a lot of precision to grab an insect when it's flying. You can see here a bat, carnivorous bat, eating frogs. They can also fish. So they have these enlarged legs. The hind limbs are enlarged, and they have these claws that they use to grab the fish from the water. And they can also feed on blood. You can see a vampire bat here feeding on a mammal. I also wanted to mention here that bats are very important for the environment because they provide ecosystem services such as dispersing seeds, which helps the forest grow, and pollinating plants, which helps keep genetic diversity or keep populations viable. And this is a good example for this is the agave plant, which is the plant of which tequila is made from. So if it wasn't for the bats, these plants would probably not be pollinated at all, and we wouldn't have tequila. So yeah, and they also are good controllers for insect populations, and they could save farmers and industries a lot of money annually on pesticides and pest controls because they feed on all of these tiny insects that could be pests for many crops. Here we have different examples of where bats like to roost. We have here on the left upper corner a big colony of bats roosting inside of a cave. We have another colony here in bunkers, so they also roost in man-made structures. You can find them under bridges as well. You can also find tree roosting bats in the forest, in holes made by other species or underneath the bark. And you can find them as well underneath the leaves. They chew on the sides of the leaves, on the center of the leaf, and the leaf kind of falls and it protects them. And then you have these impressive bats that roost inside carnivorous plants, and it's like a symbiotic relationship with these bats bringing some nutrients when they defecate inside of the plant so that the plant can insert those nutrients later on, and it provides shelter for the bat at the same time. And you have this amazing picture of flying foxes roosting all together in tree, which you might already be quite familiar with since they live here in Australia as well. Now, something very relevant for this project, and I would like you to see it's like a rough sketch of the life cycle of a bat in temperate areas, because it doesn't work for the tropics, right? And this is something that has been adapted for the Southern Hemisphere, so the months can match with the seasons. So we have winter starting around May, and it's the season that we are interested in studying, where bats hibernate. And what they do is they enter this state of torpor where they drop their body temperature, and they save energy by not thermoregulating. And that helps them go through the winter without having to eat so much, because winter is a very harsh time since there is not a lot of food in the surroundings, because insects have a threshold of activity depending on temperature as well. There could be mating during the winter, but yeah, usually it kind of changes depending on the species. Then in the spring they emerge and they go to the maternity caves where the offspring is born after finalizing all the process of gestation. In the eastern bendwing bats, which is a species that we have been studying, the gestation period happens throughout the hibernation really slowly, and then it speeds up at the end because they get pregnant sometime before the winter. And then we have the period of lactation and weaning, and the cycle starts over again. So bats are species that are very dependent on their environment and on the seasonality. If you want to learn a little bit more about bats, this is a set of books that I would recommend you to check out. This is a great book from Merlin de Tuttle, and he is one of the researchers that has published most of the bats pictures that you can find online. These pictures here are all from his authority, and all the pictures that I have included in the first part of the presentation are from Merlin Tuttle. Here is a big compendium of the information about bats taxonomy and ecology, distribution maps, everything about bats you can find in the Mammals of the World. It's a handbook. It's a very nice edition. If you're interested, I would recommend. And then you have the local editions. You have Bats of Australia, and you have Mammals of Australia, which has a nice section about bats. That's very complete. And you can also check out the website Aus Bats from the Australasian Bat Society. All right. So this was my part. And now Nikki is going to introduce the
Thank you,
coal loader.
Thomas. Yeah, it's a wonderful introduction for the bats of the world, and hopefully you guys kind of appreciate the diversity and how many species of bats are in the world as well. And so now I want to go a bit more local to the bats of the North Sydney area. So a lot of you folks might be familiar with the coal loader region, right? It's a really nice place to go for a walk, especially in the afternoon. And you can see here, there's a lot of diverse habitats that bats can utilize here, which is a very nice area to not only to capture insects, but also to enjoy the view as well, right? For these bats. So you've got forest habitats along the outer areas of the coal loader area. So where forest bats can go forage. But if you also notice as well, there's a series of tunnels just underneath the coal loader area. And that's where a lot of the cave-dwelling bats like to roost there as well. So not only that we have tree-dwelling bats, we also have cave-dwelling bats that utilize these urbanized spaces and they tend to like to hide in mines, underneath culverts, and under bridges as well. So it's a really great habitat system for these bats to live in. And it's great that North Sydney has such a really beautiful place for these bats to forage in as well. Thomas, can you go next?
Yep.
So in Sydney, there are around 17 microbat species that are known to occur in the Sydney area. And 7 of those have been found in North Sydney. There's quite a few bat species in the North Sydney area, and this was done by Leroy Gonzalez and Bradley Law back in 2013. And what Thomas is showing you now is a couple of different species that are known to be found there. So I'll just go through a few of them because they're quite interesting to look at. So the first one on the top left is the Gould's wattlebat. It's a common forest-dwelling species. It's widespread across Australia, and it's an important insect-eating bat. Because of their widespread nature, they love to eat insects, and they'll just gobble them up easily. So they're very good species to control for insect pests, if you're worried about mosquitoes, for example, during the summertime. On the bottom left is a species that we'll be focusing a lot for the rest of the talk. It's the eastern bent-winged bat, and they are found in the coal loader tunnels, which is where we did our study in. And they're a really cool bat species because they only live in caves, so they're not found living in trees and other these types of roosts. So they love living in caves, and that's where we've done most of our studies in, including coal loader. And they're considered threatened as well at a local level. So their populations are declining, likely due to habitat destructions and damages to their roost areas. On the middle graph, we have a eastern free-tail bat, and these bats are unique in terms of their ability to travel long distances. So these are really good at migrating across Australia. So during the winter and summer months, they will migrate between feeding grounds and their maternity sites. Instead of hibernating in roosts, instead they will just migrate somewhere else where it's more favorable for them. So if you're around, especially during the summer months, you might be able to hear these bats around North Sydney. We also have the large-footed Myotis. These are also considered fishing bats. And the reason why they're considered fishing bats is they have these large webbed feet that they use to scoop the water as they travel through the water surface and grab onto any fish or crabs, crustaceans that are surfacing the water, and they will feed on them. So that's why they're called fishing bats, because they can use their big webby claws to scoop up any prey that are on the water surface. So they are water specialists, if you want to view it in that respect. And there are some bats with really pointy noses as well. So we've got the yellow-bellied sheaf-tailed bat. It's also a migratory species as well, and it loves to travel above the tree lines hunting for food. And they have a very pointy nose as well. So these are some of the diversity of bat species that you might find around the North Sydney area. But have you noticed a lot of these are considered threatened or listed as threatened in at a local scale? So a lot of our bats are potentially vulnerable to external influences. For example, diseases that are coming from overseas. And this is where our main study focuses on now. So Thomas, can you go on to the next slide for me? And this is where we introduce white nose syndrome. So this is a fungal disease caused by the fungal pathogen. We call it Pseudogymnastes destructians, and for this purpose, we'll just call it PD for short. So this fungus PD was first detected in New York. So it originated from Eurasia, and then it got accidentally introduced into North America. And the first detection was in one of the caves in New York. And ever since then, it's been spreading out across the western areas of the US. And you can see on this graph here how fast it spread. So by 2021, it's basically spreaded most of North America. And if you go next, Thomas, it's basically has impacted or decimated, destroyed a lot of hibernating cave-dwelling bats. And the reason why we point out hibernating, as Thomas mentioned, during the winter months, these North American bats will tend to hibernate during winter. So they lower their body temperature down, but by doing that, they also lower down their immune system. And one property of this fungus is that it likes to grow in cold areas. So during hibernation, the fungus will grow on the bats, and it will interfere with the bat's skin, essentially makes it itchy for the bats. So they will wake up more often from that. But because they're waking up, they have to use energy to wake up, right? And so they in the end, they will lose body fat because of that frequent arousal from being infected by the fungus. And we presume the reason why a lot of these bats are dying off, some populations have 100% decline in North America, that they might be declining because of that increase in winter energy use from being infected from these fungus. Now, how the fungus is transmitted between bats is through direct transmission. So the bats would have to either be in contact with an infected bat or through the environment. So these are called fomites. So any kind of environment that has the fungal pathogen, if the bat touches it, it will get in contact and get infected by this fungus. So why is this important for Australian bats? Well, there have been several attempts to figure out whether this fungus has invaded the southern hemisphere. So in South America, especially in Chile, people have tried to swab bats and try to detect if the fungus is there, and also in southern Australia as well. And so far, it has not been detected in the southern hemisphere, which is a great sign. From what we presume, it hasn't arrived in the southern hemisphere yet, especially Australia. But there is a risk that it will arrive in Australia. So Thomas, if you click on next. So Australia is particularly at risk because in 2016, there was a risk assessment workshop done by various conservation agencies. And what they concluded, they published this in a paper in 2019. What they concluded was that it is very likely to almost certain that this fungus PD will enter Australia, and it's likely that the bats will be exposed to this fungus within the next 10 years. So this is of great concern. This was published in 2019. It's already been half a decade already, and the likelihood of this fungus arriving in Australia is probably almost, very likely. And so there is some agency to understand, well, if it does arrive in Australia, how will our bats cope with this fungus? And will our bats survive, deal with this fungus okay? So we were, well, our supervisor, Christopher Turnbull, was awarded an Australian research grant called a linkage grant. And we have a lot of different collaborators on this project. So the main investigators for this project is Western Sydney University, which consists of Chris, Justin, myself, and Thomas as well. But we also have people from Taronga Zoo, the Orthoride Institute as well, Lindy Lumsden, who is a very predominant bat researcher in Australia. University of New England as well, Fritz Geiser, University of Melbourne, Jasmine and Anna, who are working on the immunity side of this project. Because this is a very big project, we can only focus on small aspects at a time. And what we're interested in is the wintering biology of these bats. And we also have an expert as well from Canada, University of Winnipeg, Craig Willis, has done a lot of research on the white nose syndromes that are happening in Canada. So he's providing expert guidance for us on how our research can be applied. And, he can provide firsthand knowledge on what's happening at the forefront of Canadian research there. Now, we have a whole bunch of partners as well that have contributed in some way in terms of either financial or physical support or intellectual support as well. So Australasian Bat Society, Taronga Zoo, Zoos Victoria, the Australian Speleological Society, Wildlife Health Australia, the New South Wales Government, the Australian Government, and Victoria State Government. So there's a lot of institutes that have invested their time and effort into this project and that are very interested in what's happening and the outcomes of this project, right? So how are we going to achieve this? We have 4 main aims that we want to get out of this project. 1 is to look at the exposure. 2 is the sensitivity of the bats if they are infected. 3 is to develop vulnerability models to examine how vulnerable these bats are to the fungus. And 4 is to provide some actions that we can use to be enforced from these research. So for exposure, Thomas, if you go next for exposure. So for exposure, we want to look at the pathogen traits. So where the pathogen could potentially grow if it did occur in Australia. So the habitat suitability, essentially. For the sensitivity side, how sensitive would the bats be if they were infected to the fungus? And so, as Thomas mentioned before, their hibernation biology, essentially what they're doing during hibernation, during the winter months, is important for predicting how sensitive our bats will be to the fungus. And we can use this information to create vulnerability models, right? So we can predict where in Australia these bats would most likely be sensitive, and we can establish some population monitoring efforts for that. Some of the actions that we can get out of this project is targeted biosecurity. So I mentioned before that if we find certain areas, for example, the Blue Mountain areas of the Sydney region, for example, might be particularly susceptible to the fungus, well, we can advise on targeting research efforts in that area so that the bats have a better chance of surviving the fungus if we targeted that research area. We can also provide this information for cave access times. So if the bats are particularly sensitive during certain times of the year, for example, the winter months, we can provide cave tourists or cave management information about when is it appropriate to go into the caves, to enjoy themselves. And also because we're looking at the bat health as well, so the immunology, their wintering energetics, we can provide information on their health as well to veterinary people or people who are looking after bats as well. So let's provide some progress on what we're actually doing to achieve these aims. So we've got a couple of sites across New South Wales that we've been focusing on. So the color blue is the karst area. So basically these are cave regions where the bats are roosting and where we've been focusing our efforts on. So we've covered a huge area, up all the way up in Mount Capitar and the Kempsey areas in the northern New South Wales, all the way down to Kosciuszko National Park, where Yurrungabilly and Coolamon Plains are. So if you've been on holiday and been to some of the caves there, there's spectacular caves up in the Kosciuszko National Park areas. So for each of these locations where we looked at these bats, including North Sydney as well, we've done a couple of things. So the first thing that we've done is cave surveys of hibernating bats, go inside the caves or the mines to just look at where the bats are roosting, right? And we actually count how many bats there are. We also take swabs and go inside and swab the caves and the mines for PD. And so we currently have the samples. We haven't processed them yet because we're still currently doing these surveys. And so we'll be doing that at the end of our project, and we can determine whether PD or not has arrived in the New South Wales region because no effort sampling has been done so far. We've also looked at the cave microclimates as well. So we want to model the suitability for PD to grow in these caves, right? So we need to measure the temperature and humidity, which is what PD is dependent on in order to grow. We also put acoustic detectors out in these caves so we can monitor their activity. So these acoustic detectors detect sound that are produced, these high frequency sounds that are produced from these bats. So the higher the activity, the more sounds, or the higher number of sounds that are being produced, and vice versa. So we can determine activity of these bats during winter based on these acoustic detections. And we also have some time-lapse photography as well. So we put some camera traps inside the caves. These are no-glow infrared cameras, so they have no sound when they're taking photos, and they're also infrared as well, so they don't produce any light. So they don't disturb the bats at all. And so look at how often these bats arouse from hibernating. So we've done that for all the caves. And I'm just going to give you a few examples of some of these caves that we've been into. This one particularly was a difficult cave for me because I'm not a caver. So any kind of vertical component in caves is very new to me. Thomas, though, is a very experienced caver. So I took photographs of Thomas going up and down cave systems, and particularly in Drum Cave down in Bungonia. This one has a 50-meter drop down the cave, which is quite scary, but he did it with no hassle at all. And here he is in the photo surveying bats right there. Now, can any of you see where the bats are roosting in this cave? Does anyone want to give a thumbs up whether you can see the bats or not? So Thomas, yeah. So here are the bats. They're right on the top right corner. And so this is where Thomas has to go and try to count them or take photographs of them in order to get numbers and get the temperature measurements as well. Some of the caves that we have to go through require going through streams or tight spots as well. So they're often not a casual walk through caves. Most of them are, but some of them requires through squeezing through tight areas. Thomas, if you want to play a video, this is Thomas taking a video of me squeezing through one of the tunnels inside Mammoth Cave, and this is in Jenolan. The Jenolan is one of the famous cave systems in the Blue Mountains, and there are a lot of bats there. So this is an important site for us to conduct our research, but in order to get into where the bats are, we need to go through some tight spots there. Some of the cave systems are easy to go inside, but they require long hikes to get to. Now, because we don't want our equipment getting stolen by people, they either have to be fenced off, or we have to go quite far where, if someone really did want to steal them, they have to put in a lot of effort to do that. So some of the cave systems in the Kosciuszko National Park require 2.5-hour walk to get there, which is a beautiful scenery to look at as you're going through, but it's a bit of a hassle when you're carrying equipment with you to get to the caves as well. Some of them, they'll also require river crossings. So if Thomas goes into the next, if you click on next, oh, go back. If some of them, we actually have to cross rivers to get into the caves. And because this is done during the winter months, it's very cold up there. It will snow at times. And so the water is at freezing level. So luckily we have some inflatable rafts to help us cross the river to get into the caves. So here are some preliminary results that we have that we've collected during the winter months last year, and we will repeat this year as well for some new caves. So the first one was looking at exposure risk. Basically, where can these fungus potentially grow in? So are these caves that we've sampled suitable for PD to grow in? And if you look at the graph on the top left, this is the growth potential of PD for temperature. So if you notice, there's a curve going on here, and their optimal growth temperature is around 13.5 degrees, but they can grow between 11 to 16, 17 degrees. They can grow quite well in those cave conditions. So that's how cold they'll have to be to grow, but they can potentially grow anywhere between 0 to 20 degrees. With humidity, though, they love to grow in very wet humidity environments. So anything above 70%, they will grow in that cave system. 80% is their optimal, and anything above that, they will grow. So let's look at some of the cave temperatures, as Thomas pointed out. Coal loader tunnel is on the bottom left, if you want to use that as an example. All the others are caves. So if you have a look, the cave temperatures range between 5 degrees to 20 degrees. 5 degrees, you can get some really cold caves in, for example, Cormorant Cave is in Kulaman Plains, which is a very cold site. So you can get some very cold caves up to Yesaba Cave on the top left corner there, which is one of the northern caves in the Kempsey area. So there's a big range of caves there. But if you notice, the bats tend to roost in temperatures between, okay, let's say 10 degrees to up to 15 degrees, which is ideal for PD growth. So if PD does arrive in Australia, these bats are roosting in areas that PD can grow very well in. So that's a worrying sign from a temperature standpoint, including coal odor. What about humidity? So humidity is a different scenario. A lot of the caves, they have very high humidity, which is also bad if the bats are roosting it. The bats love humid caves because when they go into hibernation, they're saving energy, but they will lose water across time because they're not going out to drink water, right? So if they live in a very humid environment, they don't need to wake up as often to go and drink water. So a lot of these caves that they tend to roost in are very humid environments. But the great thing about coal loader is that it's not a very humid environment. Because it's a tunnel, it's open-ended on both sides. And so there's a lot of wind flow going across both sides, which means that the humidity doesn't build up as much there. And so the bats that roost in those caves, it's likely that PD won't grow very well in those caves because the humidity isn't high enough for these bats. But for the rest of the cave, they will do well. So if you press next, Thomas, there is a high exposure risk to PD for these bats in these cave areas. So it's not a good sign so far. Okay, to go next, Thomas. We've also got some camera trap recordings that we've done, and we're going to count these bats and how often they wake up as well once we start processing the data. But here's just an example to show you guys how we've done this. So the left side corner, left side of the screen here, we have a map of the cave. The entrance is on the top, so we basically go across the entrance and walk all the way down to that blue highlighted area. That's where the bats are, and we place the camera trap there, pointing at one of the cave walls. So if you click on next, Thomas, the folks here can see a video of how often these bats are waking up. So these little blotch screens that you can see here are where the bats are roosting. And we also have time, and these photographs are taken in hours as well. So you can see the time here, and a lot of the bats that you can see roosting in this cave system, they love to stay in the same spot with a few individuals moving in and out at times. So it's great to see that these bats are mostly hibernating, and some individuals are arousing, waking up, moving to different spots, maybe going out to feed for a bit and then coming back if they think it's good. We also have another aspect that we're looking into, which is the sensitivity of these bats to white nose syndrome. Now, because this aspect takes up a lot of effort, we can only focus on a few caves at a time. So for the caves that we did look at, if you click on next, Thomas, we considered a warm site, which is the Kempsey area, and a cool site, which is in the Jenolan Blue Mountain areas. We captured bats in those areas and measured their body condition, essentially their weight and their forearm as well, to look at their body condition, and also the thermal biology as well. So Thomas will talk a bit about the radio transmitter work that he's been doing and some of the results there. But what I'll present to you now is the body condition that we got from those two sites. So if you can clone next, Thomas. So now we're looking at a bit about the sensitivity risk of these bats. And this is in terms of how much body fat they're losing over the winter period. So pre-winter is around May, just as winter is starting. Midwinter is July, right in the middle of winter. Late winter is in August, and early spring is in September, near the end of September. We've got the cold group at the very top there, the Jenolan group, and the warm site, which is the Kempsey area, Yesaba Cave, on the bottom there. Now, there's a clear trend that you can see on here with the body mass of the Jenolan group. The females and males, both of them, have a huge decrease in their body weight, so in their weight, from pre-winter, at the beginning of winter, to the end of winter, right? In fact, the females are losing up to 30% of their body weight across that winter period. So this cold site that they're living in, the females are losing a lot more body mass because of the cold conditions that they're living in. Whereas in the warm site, they don't lose as much body weight, especially the males don't. The females do lose a little bit of body mass during the winter period, but nothing compared to the cold site that they're looking at. So how does this compare to other bats in the Northern Hemisphere that are experiencing declines from white nose syndrome? So we can give an estimate of how sensitive they are, how much fat do they lose during winter with other bats in the US. So on the right-hand side graph here, we've got winter body mass loss in the top, in the y-axis. So the higher the number, the more mass they are losing, and the annual surface temperature of that particular location is on the x-axis. So 0 is very cold seasonal environments. This is areas like the Canada areas or the temperate rainforest of the US, up to 28 degrees, which are subtropical, tropical areas. And you can see a trend here, whereas the colder the climate is, the more mass they lose over winter. And I highlighted in blue where the owl bats that we sampled fit in with these graphs here. So Jenolan has a mean annual surface temperature around 13, 14 degrees, and they lose body mass similar to North American bats that live in similar conditions, similar climates as well. And same thing with the Yesaba bats as well. So this is interesting. They have a very similar energetic use during winter with North American bats, which tells us a bit about how sensitive they could be if they are infected with PD. So we go on to next now. Oh, so they have similar winter mass loss to other bat species, essentially. So we go on to next. So this is where Thomas will now talk about his radiotelemetry work on how much, essentially, how often they go into torpor or hibernation, and how often they arouse from that. So I'll leave it to you now, Thomas.
Thank you, Nikki. That was a great presentation. Now I'm going to talk a little bit about what I've been doing with my temperature sensitive transmitters. First of all, you have here a picture of co-loader actually, with the harp traps placed at the entrance. We at the beginning we actually wanted to use co-loader as our warm side, but because the bat population there is quite variable, you can see changes in numbers very drastically throughout the winter because they are so active because it's warm. So it was not a suitable site for this particular part of the project. So we would have to capture all the bat, well, a group of bats, and I would put transmitters, 9 transmitters on the back of the bats. You can see here in the middle in the picture of the middle, this bat has glued a transmitter on the back. We just trim a little bit of the fur, add some adhesive, and put it there. Wait for a couple of seconds, and then they're good to go. Then when we release them, this is a picture of the receivers that we use. They have been made here in Australia by someone that our supervisor knows very well and has been helping him produce these fine pieces of equipment that we use to get the pulse intervals of the transmitters. And what that gives us is the skin temperature, which is what we want to use as a proxy to measure how they are using torpor. If it's pulsing faster, then the temperature is higher, and if it's pulsing at a longer interval or slower, then the temperature goes down as well. And we have those equivalences, and we can make the calculations. And with these receivers, what we're doing is collecting all that data. So on the next slide, we can see after capturing the bats, how we deploy them, how we release them, sorry. And on the right side, you can see me tracking the bats back inside of the cave, and listening for the transmitters and see if all the equipment is working, how many bats we have present in the cave, and making sure everything is in working order. Here we have on the left side a picture of me with an antenna deployed on the ground next to me, and taking notes and downloading data. So we have to visit the caves every fortnight and collect the data and change batteries because these batteries run out quite fast. And we always try to do this under red light to avoid disturbance to the bats, and all the equipment it's placed in a different chamber to avoid this disruption of the bats. And the antennas, we have long cables that connect to the antennas that are at the chamber where the bats are roosting, and which is actually receiving all these signals from the transmitters that we have deployed. And we found something really interesting on this study, and it's that there is a difference in the use of torpor in between the warm side and the cold side. As you can see on the left, on the big plot on the left side, on blue we have our cold side, which is Genolan, and you can see that at the middle of the winter, which is August, July, late July, August, we can see a trend where they use torpor for longer periods of time, up to 6 days. It's the maximum we found in this study. There has been a previous study from a master's student that has found torpor bouts of up to 14 days, which is pretty long. And then when we compare these to the warm side, we can see that they barely use torpor for more than 1 day. There is one case where they use torpor for 2 days, but they are very active at the warm side, and they go out of torpor and they fly out, they look for food, and they come back. So they don't stay in torpor for long periods of time, which is a very nice result. And it can reflect how many resources they have available and how they adapted to different types of environments within their distribution, even though they are the same species. You can see here on the lower right side, we have the top about duration overall, and you can see how it's different between the warm side and the cold side. You can see that on average at Yesaba, bats use torpor for shorter periods of time. And then the Genolan side, which is the cold side, shows that they can use torpor for up to 24, 48, 72 hours. And you can't see it on here because the background is black, but we had some few peaks there at 144 days and 120 days, which is up to 6 days, as I mentioned before. And then on the upper right corner, we have arousal time between sites. And that's telling us at what time they're waking up when they wake up. And usually we can see here that at Yesaba, they wake up at different times all day. But there is a peak of arousals at sunset. So 25 minutes, 30 minutes after sunset, all the bats tend to wake up. They become more active. And sometimes they go back into torpor, or sometimes they go out. This is some data that we collected just yesterday from CoLoader. And you can see how the temperature changes throughout the year. So we have the external temperature, which is more variable. We have a gap here of data because we don't have information for the next months. And we can see that it also mirrors the temperature inside the cave at very shallow distances from the entrance at the middle of the tunnel. It's very similar, the trend in temperature. So, which means that, as Nikki mentioned, the tunnel has a lot of impact of the external environment, and there is a lot of variability, and that's why the humidity is not as high as we would expect at the areas where they roost. I also noticed there was a change in bat numbers. During the winter, we only found up to 100 bats roosting inside of the tunnel. And yesterday, when I went to CoLoader, I saw higher numbers of bats. They were roosting in bigger numbers. I found about 400 bats inside the tunnel. And we have here humidity, as Nikki mentioned before, is not as high as in the other places, and you can see how variable this is, and because of the external influence in the tunnel effect, and the wind goes through and takes all the humidity. Okay, now to finish, we have here our website, the Bats Lab. If you want to look for more information, you can visit our webpage or contact us at Nikki's email. And you can also find more information on Australasian Bat Society website, Wildlife Health Australia, and the US Fish and Wildlife Service for information related to the white-nose syndrome. With further ado, thank you everyone for joining this session, and I think we're open for questions now.
Oh, thanks so much for that presentation, Thomas and Nicholas. It was really interesting. I think we've got a few questions there. Maybe you'd like to quickly scan through and answer any that you think would be interesting.
Yeah, sure thing. I think we can go from the very start. I think there was a question right at the beginning of the presentation from Emma. When you mentioned vaccination against rabies transmitted by bats, does it mean that the normal 2-part rabies vaccination, such as available in the US, was there special vaccination for working with these bats? No, there essentially works for the rabies here in Australia as well. So we have a different virus. This is it's called the Larissa virus that is found in Australia, and that differs from the North American variant. But the vaccination protects you from both. So if you have the vaccination, the rabies vaccination in general, you will be protected here as well. I mean, it's advisable, just don't get bitten by a bat in general. But, if you do get bitten and you've got this vaccination, you're generally safe from it. If we go to the next question. Oh, did you want to mention something, Andrew?
Oh, no, I was just, no.
Yoon said, Australian Bat second edition is no longer in print. You can borrow it from a library or download it digitally. Okay, I did not know that. I think our university library has a copy as well. So is that true, Thomas?
We have a copy at the Western Sydney Library at the Hawkesbury campus, but for the first edition, it's very hard to get the second edition, actually.
I see.
I haven't been able to find that well, I haven't looked for it at different libraries, and I certainly don't know how to download it. If you know where we can download it through a library, it would be great if it's available. Otherwise,
Yeah.
I guess we would have to wait for the next edition, which will hopefully come at any time soon.
Maybe that person who suggested that could post the link in the chat.
Yeah, which library could you find it in?
Thomas, can I ask you to stop sharing? Thanks.
So there was another question from Glenn. Why is it inevitable the fungus will come here? So Glenn, it is not inevitable. There is a likelihood, doesn't mean it is certain that the fungus will be here, but that likelihood is based on the fact that a lot of cavers aren't aware of this fungus. I'm not going to try to put blame on anyone, but Australia, even though they have high biosecurity measures, they're only for known diseases that can affect Australian wildlife. But because the white nose syndrome is relatively new compared to other known diseases, the biosecurity measures involved with preventing PD from coming here is virtually low. So it's hard to kind of predict, when it will come here. These are based on expert panels that have assessed this and account for biosecurity measures, how often tourists come here, specific people who are going to caves, bringing in equipment from overseas, going in here, that might influence the likelihood of this fungus coming in here. So I won't say it's inevitable. It's just a high likelihood.
So it's not the bats, is it? Like the bats can't microbats can't migrate that far.
The microbats can't. Not fruit bats. The flying foxes can, but there has been no cases of flying foxes with PD in, for example, the Southeast Asia. And because Southeast Asia is very warm, it kind of provides a thermal barrier for this fungus to cross over to anyway, naturally. So the likelihood for Australia's case is through human intervention.
Yeah. Something to note, well, even though flying foxes fly long distances, I don't think this is something like crossing the Pacific Ocean. They don't fly as long distances to accomplish that. But
they have been known to. It's not unusual, but it's not a normal migration pattern that they have crossing continents, but it is possible.
Yeah, and it's possible that the fungus already arrived here, but it didn't infect bats. That's something we won't know because it's very hard to keep everyone on check and sample for all their equipments. It's something that you don't see. Its fungal spores are very tiny.
Yeah.
So it could happen that the fungus arrived a long time ago and it never infected bats. And it could be that at some point it will, and that's unsure. And that's why we're interested in how vulnerable bats actually are to this fungal disease if it was to come and infect them.
Nicholas, concerning our site, the coal odor, from the information you presented, it looked like the humidity wasn't a problem, but it's within the temperature range. Like how how vulnerable is that population of microbats to PD?
Probably not too vulnerable, because it's not just temperature that they require, that the fungus requires, it's the humidity. So the temperature is basically how fast the fungus could grow, and the humidity is whether or not they can grow. So if the humidity is not there for the fungus, it doesn't matter what temperature that they're in. It's hard for them to grow if it's just too dry for them to grow in, right? But note, if it rains a lot, it will increase the humidity in the tunnel, and the fungus can grow on the bats. But because of so much variability relative to other caves, how fast the fungus grow might be lower than other cave systems. And because bats there are quite active during winter, that we noticed, including Leroy's work, because they're waking up more often, they're likely not going to develop the fungus as quickly because they're waking up more often. It's when they're in torpor, as Thomas mentioned, the longer they are in torpor, the more the fungus can grow on them.
Because they're
Yeah.
cleaning themselves all the time?
No, it's because when they arouse from torpor, their body temperature goes up way above what the fungus can grow, right? About
I see.
37, 38 degrees is their normal body temperature, right? But when they're in torpor, that body temperature goes way down to the environmental temperature. Actually, Thomas, in the future, if you like, you can give an example graph of, when they're aroused and when they go into torpor, the temperature difference.
Yeah.
You know those temperature traces? Yeah. Maybe you can show that next time.
Yeah, that'd be great. Yeah.
There's a biosecurity question there from Michaela. What measures would we need to prevent it getting here?
What measures? So it's not really I'll say what can be done. What we can only do is recommend information to them on where it is most likely, which areas are most likely going to be particularly vulnerable. And what they will do with that information is up to them, whether or not they want to bump up the biosecurity measures. So, either preventing, it's very hard, once it comes here, it's very hard to prevent the fungus from spreading to other systems, because once it's on a bat, if the bat travels to another cave, it's going to infect other bats, right? And it will continue spreading in that fashion. And what we're trying to figure out is how connected. So one of the projects that we're looking at is how connected these caves are with each other. So essentially, it will tell you how, if a bat does get infected, how likely is it going to spread across different cave systems? If it's an isolated case, if it's a meta-population case where it could spread potentially anywhere, that will also need to be determined as well. So in terms of preemptive measures, one thing that can be helpful, if you ever go into a cave, or even mines as well, clean your boots after you leave the cave or mine. So for example, if you go into coal loader and there might be PD in there, fingers crossed it doesn't, clean your boots after with disinfectant, whatever disinfectant that you have, just scrub off any dirt on your boots. And when you go to the next area that you're interested in, you're less likely to transmit that pathogen over. So it's just keeping a very good hygiene protocol overall when you're going between sites.
Also, if you're aware of a colony of bats that it's roosting at some place during the winter, it's advisable to stay away from that colony, because the presence of people in those areas will definitely affect the bats and will make them wake up and use up all the energy that they have saved for the winter if it's very cold side.
I know with Phytophthora, bush regenerators use a methylated spirits mix, and we spray that on our shoes to stop the root rot spreading around.
Nice.
I believe that could work. I did read a paper on different types of disinfectants and how effective they were. Essentially, most of them were effective at killing, I think, at least 90% of the fungus. So most common household disinfectants would work. I think the most effective was ethanol or those veterinary disinfectants, F1 or F11 or something. Yeah.
Another way of sterilizing is washing
F10,
your
that's it.
clothes at 55 degrees Celsius for 5 minutes.
That's right. So as we mentioned before, the fungus does not like warm environments. So actually soaking, for example, if you accidentally fall onto the tunnel and get your clothes dirty, soak it in very warm water. I think what you said, Thomas, at least 55 degrees, right? For
For
5
5
minutes.
minutes.
Yeah. So the 5-5 rule or something. Yeah. Will clear away the fungus. So if you don't want to use harsh chemicals, you can just soak it or boil whatever item that you have that you think might be infected.
Yeah, they
To
might
get
be a
rid of the
bit
fungus.
smaller. They might end
Yeah.
up being a bit smaller afterwards.
The clothing's right. Yeah. That's right.
What other questions do we
We
have?
There
had
seems
a
to be a lot of questions. Thomas,
yeah,
do you want to pick some?
we had a question from Jan. He asked what model of bat detectors we were using and if we were using active and passive detectors.
Yep.
We were using only passive detectors. We were using SM4s from Wildlife Acoustic, bat detectors, full spectrum, and we were recording every day, well, every night, from 30 minutes before sunset and 30 minutes after sunset throughout the winter. And we were changing batteries and SD cards every 1 or 1.5 months.
Yep. Andrew, you also had a comment about cold conditions. Did our presentation kind of answer that, or do you want some further elaboration on that?
Oh, sorry, did you ask me a question?
Yeah, you said in your in one of the comments when you
Oh,
say
yeah, I
PD
think you
likes
covered
cold.
that already. Yeah,
Okay.
yeah.
All right. Let's see if there's another one.
There was interesting. Avril has asked, can PD affect snakes and reptiles and amphibians?
So there was a similar fungus called chytrid fungus that has been affecting frogs in Australia, and they do similar things because they like to live on the skin and eat the keratin layers on the skin. But PD seems to be specific to bats only so far. To our knowledge, it has not been tested for reptiles and amphibians, and this is because we don't find many reptiles and amphibians in caves in Australia, right? So it's even in the US as well, there's not many. And if there are, it's very hard to find them and test them. I think, I'm not sure, but to our knowledge, right, Thomas, that I don't think it's been tested, and I presume it doesn't affect reptiles and amphibians.
Yeah, so far, white nose syndrome only affects bats.
Yeah.
Not even other mammals, just bats.
Someone asked about fruit bats.
Fruit bats,
Flying
flying
foxes.
foxes.
Yeah, how
More,
serious is the threat to our flying foxes?
yeah. Do you want to answer that, Thomas?
Yeah, sure. So because flying foxes don't hibernate like bats do, and they don't roost under these very humid and constant environments, it is almost impossible that they will get infected with white nose syndrome. So it's not a threat for the flying foxes. It's only a threat for cave roosting bats that roost in these very particular cold conditions and where they hibernate for long periods of time and they have these high humidities.
Thanks.
What I was going to ask Nicholas, what are our friends in America are doing? How are they responding to white nose syndrome? Are they able to do anything to control it?
It's hard to it's hard to control once it's already so widespread. The best things they can do at the moment is follow decontamination protocols to decrease the risk of spreading it further from human, any kind of human influence of spreading it, reduce it as much as possible. What they've also done as well is, because these bats are particularly weak in winter, especially if they're infected by PD, they will avoid going to the caves at all during winter, to decrease that risk of them waking up, without needing to, right? Because there are signs that there are populations recovering. Those that survive tend to have strategies that better allow them to survive the winter, even with PD infection. For example, choosing areas of the cave that might be less suitable for PD growth, or maybe moving to areas, different cave systems that allow them to, if they do wake up, allow them to go and feed and build up that fat reserves. So having any kind of mitigations from human side to reduce their energy use during winter will help them, because apparently there are populations that are recovering on their own. So we don't, we kind of want to leave nature to do its thing to kind of recover themselves, let natural selection do its thing while we can try to reduce that risk. So that's what they're trying to do on that side. But they're also trying to find ways to get rid of the fungus in terms of, I believe, vaccinations for bats.
Wow.
I don't know how successful that is. This is all at the moment very preliminary, all done in the labs. I think I don't know how successful they are as well, but that's my knowledge on what they're doing to help the bats there.
There's a related question from Emma. Have wildlife rehabilitators been successful in treating affected bats?
In the US, yes, I believe so. So when the bats are infected, the important thing is that they need to get enough energy reserves to survive winter, right? If they're with a bat rehabilitator, the bat rehabilitator will feed them, even scrub off the fungus as well on their skin, and help them recover in that way. So even if they are irritated, if the bat is able to fight off the fungus themselves while getting enough nutrition, they can do it themselves. That's why in the wild you'll see bats still survive, because they're not using up all the energy reserves during winter. They either have some way, managing to find insects during winter, or finding a place where the fungus doesn't grow as much to be able to survive. So rehabilitators will just offer them more food. The bats will tend to survive better.
Oh, great. I think someone said, Michaela said the F10, that was probably your
Yeah.
antiseptic there, yeah. All
That's
right. Have we
I think Avril asked if there were any studies conducted on resilient bat populations.
Absolutely, there has been, and there are differences in their behavior and their immunity as well, to compared to bats that have declined. So, for example, PD is found in Europe, right? Europe and Asia, but the bats don't seem to be declining there, and there are climates there that are suitable for PD to grow. One thing that is happening there is that the bats have co-evolved with this fungus, so that when the fungus is on the bat skin, the bat's immune response is different from the bats in North America. The bats in North America are going into overdrive, which is why they are waking up and trying to get rid of the fungus, while the bats in Europe seem to be dealing well with the fungus. They can effectively get rid of the fungus, or at least manage with the fungus, without needing to wake up as often. How they do that requires a bit of underlying physiology, which probably is a bit too detailed here to explain, but there are differences.
Thank you.
Well, if there are no more questions, I think we will wrap up the talk. And a big thank you to you, Nicholas and Thomas. I didn't mention that they very kindly invited me to go along and do some of the survey work in the Collota Tunnel which was really interesting to see how it's all done. So I'd it'd be great if I could do that again with you sometime, and we'll keep track of your work.
Oh, thank you, Andrew. I appreciate it. Definitely, whenever we go back, we'll give you an invite. And 400 bats, Thomas, that's impressive. Wow.
Yeah, it was, and they were clustering not inside the shafts, but outside of the shafts, in between the
Ah.
shafts actually, and the rooftop of the tunnel.
Yeah, that's right. That's what Leroy said as well last time. Because Andrew, you came in last time. There wasn't that many bats, so
No.
to you, it probably didn't look that impressive. But from what Thomas said, they're probably overcrowded in the shaft, so they're moving towards the sides as well.
Yeah,
Oh.
actually the shafts were empty.
Oh, okay,
I think they
interesting.
just prefer different conditions because maybe it's too warm and they want something colder and they have air coming through.
Ah, okay. Right.
Yeah, and they were very active, very chatty.
Oh, of course. It's still quite warm out here, except for today.
Well, it's great to know North Sydney still has a healthy bentwing bat population.
Yes.
All right, guys, we'll finish it up there. Thanks very much for coming and speaking with the community tonight.
No worries. Thank you,
so much for
everyone.
having us.
Yeah.
Good night, everyone.
Good night.